Life Science learners, to another installment. You guys have looked at DNA in the past. Today we're looking at the role of DNA and replication. There's a lot that we need to discuss today. So let's get out those notepads. Let's try and engage with the content. And I'm hoping that you've got some questions that you can write down and I can get to them as we discuss. So let's look at DNA and, very important, replication. It's important at this point that we have some reflection on kind of what the structure of DNA is, to look at where it's found before we get into understanding the process of replication. So I'm going to spend some time looking at a recap, and then we're going to move on to the process of replication. So please bear with me. So I'm sure you always have had thoughts about DNA and what information it codes for. So we're going to spend some time trying to understand what the concept of a gene is today. We're going to focus on the nucleus, genes and chromosomes in this segment. As a recap of nucleic acids, we're going to talk about that. And most importantly today, we're going to look at this complex process of replication. And we're going to unpack the different models. More importantly is the process, its significance, and when it occurs. So let's get straight into some key concepts as we unpack. And these are some key words that are important that I would use through this lesson. So the concept of what chromatin network is. Chromosomes, we're going to unpack what chromosomes are and how they're made up of chromatids. We're going to look at histone proteins forming an important part of a chromatid. A very important concept to understanding replication is the genes and the role of genes. And we've mentioned nucleotides as the building blocks of DNA. And we know that these contain different nitrogenous bases. We're going to spend some time just looking at DNA and comparing that to RNA. And I will mention the types of RNA and DNA, and we're going to talk briefly about mitochondrial DNA. We can also refer to complementary base pairing, especially when we look at DNA replication, and we're going to possibly get to chromosome mapping during the course of the lesson. So a quick recap of the structure of the nucleus. Remember that the nucleus is that organelle in the cell that contains the chromatin network. And if you look very carefully into this diagram, you can see that I've got an illustration of a shadow of a chromosome in there. And that chromosome contains the genetic information that's wrapped up around histone proteins. We know that the nucleus has nucleopores. It has got an inner and outer membrane, which basically controls the movement of substances into and out of the cell. It has an internal solution called the nucleoplasm, and it has a little structure called the nucleolus, which is linked to how the nucleus carries out its functions, especially in terms of cell division. So we've looked at the structure of DNA. We said that it contains, it's found in the nucleus, and it forms part of what we know as chromatin. And chromatin is actually can be arranged into a compact structure known as chromosomes, which I will illustrate to you in the next couple slides. We mentioned that the nucleus is surrounded by a double envelope, which contains 2 layers, and it has pores, which allows the movement of substances in and out. The nucleus also contains a region called the nucleolus, which is a little structure in there, which we know controls how the nucleus functions. The nucleus contains DNA in the form of chromatin, as I mentioned, and that chromatin we know is basically chromosomes that are wrapped around histone proteins in a very complex structure. It's important that we have an overview of the structure of a chromosome. We often draw this as an X-shaped structure, and it's important that you understand that this X-shaped structure is essentially DNA that's complex structure wrapped around histone proteins. Okay? And so these chromosomes are thread-like structures that are located inside the nucleus of an animal cell and plant cells. And these chromosomes contain proteins called histone proteins around which the DNA is wrapped. Okay? And obviously it's important that we understand that the term chromosome is made up of 2 words, chroma and soma, which refers to color bodies. And this is indicative of how the chromosome basically stains with different colors to produce these bright colors, which scientists have termed chromosomes, using color and body to break up that. Again, an overview of the structure. We know that DNA is a double-stranded molecule. It contains complementary base pairs. It is arranged in a helical structure, which we see here. And if we compare that to the other type of nucleic acid, which is RNA, we see that RNA is single-stranded. The location of the nucleus. It's important for us to understand the context of where the nucleus is and what it contains. So the DNA occurs mainly in the nucleus, which is formed part of the chromatin network. It is also known as chromosomal DNA. So we have DNA found in the nucleus known as chromosomal DNA. We also have DNA found outside the nucleus, and that's found in the mitochondria as well as the chloroplast in plant cells. DNA is a polynucleotide structure, guys, made up of many nucleotides. It is a polymer, and it's made up of building blocks called nucleotides. So these monomers of DNA are called DNA nucleotides. So these are the monomers. It's important that we unpack that the structure of each monomer or each nucleotide consists of three parts: a sugar molecule, a phosphate group, and a nitrogen-containing base. And now I've illustrated that here. So we see a phosphate molecule attached to the sugar. We see a nitrogenous base here, and we know that these are made up of adenine, thymine, guanine, and cytosine. As I mentioned here, as a recap, we know that these are 3 or 4 different types of possible bases that are present on a nucleotide. This is crucial to us moving forward. The concept of a gene. What is a gene? And often when we look at DNA, we refer to DNA containing genes. It's important that we understand that a gene is a sequence of DNA that has a special meaning or codes for a unique function. So that gene is made up of a sequence of nucleotides that code for a specific function. And that talks to how DNA codes for characteristics. So a sequence of nucleotides that code for a specific characteristic or protein is called a gene. And a gene is essentially the basis of the physical and functional unit of hereditary information. So this is essentially how information is passed down from one cell to the next cell during cell division, as well as from one generation to the next generation in terms of reproduction. So genes are made up of DNA. Some of these genes basically act as instructions to make molecules called proteins. So in our future installments, we will look at a process of protein synthesis. And so genes code for the information to make specific proteins. We also know that genes do not code for proteins in some instances. They code for how cells express themselves or how certain processes occur in a cell. So we're going to have to look at that as well. So a gene, if we were to basically describe that, is a segment of DNA that codes for a specific characteristic, which is referred to as coding DNA. It is important for us to understand that we have lots of DNA, but not all of that DNA codes for characteristics. It is said that only 2% of our DNA actually codes for specific characteristics. If we look at the other 98%, we refer to that as non-coding DNA. And so, as I mentioned, that 98% of our DNA is referred to as non-coding DNA. In some textbooks, we've referred to non-coding DNA previously as nonsense DNA or junk DNA. However, as scientists have understood the sequencing of DNA and the Human Genome Project, we're able to identify how the non-coding DNA seems to now have a significant role in helping the coding DNA express itself. So the non-coding DNA is important in allowing for the expression of some genes. So that's important for us to take note of. So genes can have one of two functions, guys. Genes, when we refer to them and we talk about structural genes, these carry the code for one or more proteins. So these genes basically produce proteins. But we also have regulatory genes. And these are genes that regulate processes in cells and organisms. So broadly classifying genes, we have these, as I mentioned, structural genes that code for making specific characteristics of proteins. And we have regulatory genes, which basically switch on or switch off certain processes in the cell. And that's important in us understanding how genes carry out their functions. the concept of a gene locus. And the word locus is derived from the word location. And so we know that genes have a specific position that they occupy. They have a specific length, and they're found on unique chromosomes. And so that position of a gene on a chromosome is referred to as its locus. So the locus is the position of a gene on a chromosome. We will come across the term allele, and that concept of allele is important for us to unpack as we move on. Let's expose ourselves to what an allele is. An allele is essentially a different form of the same gene. So if we think of a simple characteristic like eye color, we've got different variations in eye color. If we think of left-handed and right-handedness, those are the different or the alternative forms of the expression of a gene. So we have alleles for those different expressions of genes. Right, so in this illustration, I've illustrated a pair of homologous chromosomes. Again, the concept of homologous chromosomes comes from the fact that these chromosomes are similar in shape and size, and they basically are representing an allele for white flower. In this case, it's indicated by this purple region, and on a corresponding chromosome, the allele for purple flower. What is important is in this, we understand the concept of what is the location for this specific gene, and we use the term locus. And so essentially, by me trying to illustrate the position of a locus, I'm using this image to show you where that specific gene is found. And so remember that that gene is DNA that is wrapped around histone proteins that codes for, in this case, the color of flower. And these homologous chromosomes essentially point to structures that are where DNA is found and wrapped around histone proteins. So guys, we've looked at the nucleus, we've looked at the genes, and we've tried to unpack what a chromosome is. It's important that we understand this context before we move into the process of DNA replication. So I'm going to pause here. We're going to move into the next segment, looking at the process of DNA replication. See you in a little while, shortly after the break. Welcome back, folks, to the next segment. In this segment, we're going to look at the role of DNA and a very important process of DNA replication. So let's try and understand what is the role of DNA in a cell. So when we try and understand the role of DNA, it's important for us to look at the different roles that DNA plays. Firstly, we're going to look at the role of DNA in the process of replication, which is fundamental to our lesson today. We're also going to spend some time looking at encoding information. So what does that mean for a cell? We also know that DNA has an important impact on mutations and recombinations of genetic information. And what we also need to spend some time looking at is gene expression. Why are certain genes switched on, and in which cells are they switched on? So, if we try and understand replication as an overview of the role of DNA, we know that cells divide, and in order for cells to divide, there needs to be some process of organization. So when a cell divides, the chromosomes containing DNA strands must replicate. And another word for replicate is copy. So it's important that the DNA of a cell has to be copied or make copies of themselves, so that when we produce two cells or daughter cells, that these two cells receive a full set of genetic information. So the rationale would be that if we've got a cell that divides to produce two new cells, that these cells must have an exact copy of that genetic information. And so it's important that that information is copied so that these daughter cells have that information. And so that copying process is replication of DNA. The second role of DNA is encoding information. And if you think about the word encoding, it comes from the word coding. So essentially, the sequence of bases in a gene determine the sequence of amino acids in proteins, which essentially is an important biochemical unit of the cell structure and function. So if we go back to, or we look ahead in terms of what happens next, we know that genes code for characteristics. And so this essentially means that in order for certain processes to happen, for example, protein synthesis, that information needs to be coded for by the DNA. So we're looking at how DNA plays an important role in coding for information that makes structural components like proteins, like enzymes. Mutations and recombination. DNA plays a role in the evolution of a species. Chromosomal DNA helix don't usually interact with each other. However, through the process of genetic recombination, segments of different chromosomes swap places with each other, creating new sequences of genetic material. So we refer to mutations as what drives changes in species. The fact that we have the genetic recombination when two alleles from parents, from a sperm and egg, come together. There's a combination of characteristics, and that talks to how the genes in that combination are expressed. So we refer to the recombination during sexual reproduction, creating combinations of characteristics that are different in the offspring. We also know that mutations can randomly occur, and those mutations are driven by the process of replication going wrong. Yes, something that goes wrong that gives an individual a mutation can be an advantage, could be a disadvantage. And essentially that is what drives variation in a population, and that variation leads to some being better suited than others. And so DNA is important in that process as well. We also know that there's an understanding that gene expression is unique in a cell. So we know that we've got different cells, and in different cells, certain characteristics or processes are stimulated. Those cells need to be switched on to produce a specific protein. For example, the cells in the pancreas will produce the hormones needed for regulation of blood sugar. There are certain cells that will produce certain enzymes in different parts of the body. And these we refer to as gene expression. So let's understand that. So each cell contains a full complement of genes. So all our cells have the same number of genes. Yet, cells from the different tissues and organs look and behave very differently. As I mentioned, the reason is that some of the DNA of each cell is used to make proteins. Not all, so certain genes are switched on. So we say that DNA plays a role as a traffic cop for the types of proteins a cell will make. It does this through interactions with proteins in the cells caused only certain genes to be expressed and to express themselves. So we say that not all genes are expressed in every cell. Certain factors regulate the expression of genes in a cell. And so we've looked at the role of DNA. Now let's get to the process of understanding replication. So what is DNA replication? If we refer to the term replication, replication essentially means copying. So it's a process where the DNA in a cell makes a copy of its cells during cell division. Okay? Where does DNA replication occur? It's a very interesting question. DNA replication occurs in the cytoplasm of prokaryotes. Remember that your prokaryotes are cells that do not have a membrane-bound nucleus, and hence it happens in the cytoplasm. However, in eukaryotes, these are organisms that have a defined nucleus, we know that it happens in the nucleus. However, what is important is that regardless of where the DNA replication occurs, the basic process which we're going to look at is the same. The structure of DNA lends itself easily to the DNA replication. Essentially, when we look at the structure of DNA, which is a double-stranded molecule having those complementary base pairs with complementary nucleotides on either end, it's that structure that leads to the process of DNA replication being universal. Right. So this is an illustration of when in the cell cycle this process of DNA replication occurs. We know that a cell goes through different stages, and what is unique is that the process of DNA synthesis or replication occurs in a phase of the cell cycle called interphase. We have looked at cell division where we spoke about cells growing and dividing during mitosis and in meiosis, but we also know that the cell undergoes a process of specialization and development, and that during the synthesis stage, it will go through what we know as replication. And so that is in interphase that the cell undergoes replication. And so that's key for our understanding, is that in interphase, the cell will undergo the process of cell division. And we see this in mitosis and meiosis, and it's important that the cell is able to copy the DNA so that the daughter cells at the end of cell division have an exact copy of the genetic information. What is the significance of replication? The purpose of DNA replication is to produce two identical copies of DNA. This is essential for cell division during the growth and the repair of damaged tissue. DNA replication also ensures that at the end of each new cell receives its own copy, an exact copy of DNA. So when cells repair and regenerate, it is important that these cells have the same genetic information as the original cell that they've formed from. And so that is why it's important that DNA replication produces identical copies of these cells. So let's understand the process of DNA replication. It's important that we identify what is needed. And so there's a series of needs for this process to occur. The first in most biochemical process is enzymes. And the enzymes we're going to look at is a group of enzymes, and we refer to them broadly speaking as DNA polymerase. The focus is important that we recognize that these are DNA polymerase enzymes. We also need the building blocks. If we were to make DNA, we need the building blocks of DNA. And so it's important that we have the building blocks called DNA nucleotides. When copying or replicating, we need a reference, and that reference strand to copy from is the DNA template. So we're going to need a template to copy from. So in this illustration, I've outlined the process of DNA replication in a series of different steps that are illustrated in numbers. I'm going to talk you through this process so that we can understand essentially what happens. I'm hoping that in this illustration that we can go through each process trying to understand exactly how the DNA molecule is able to produce a new copy. So the first process in DNA replication is that the DNA will unwind and unzip. So here we see the DNA molecule. It's going through a process of unwinding and unzipping. If you refer to a zip on a jacket, a zip in a jacket will allow for you to open the halves that are combined together. Similar to that, DNA is held together by hydrogen bonds, and these bonds need to be broken up. So we have an enzyme called helicase that breaks the weak hydrogen bonds, and here you can see that enzyme illustrated in this by this arrow, which is an enzyme that breaks the hydrogen bonds holding the complementary base pairs together. And this causes the separation of the two strands of the DNA into templates on either side. So we now see that the enzyme breaks open the strands, causing a separation. What happens next is that these exposed strands that we see that have separated now act as a template. A template is essentially something we copy from. So if you take a worksheet to a photocopying machine, and if you place that over, the photocopy uses that as a template to make more copies. So we've got two templates that are now serving as a reference strand for us to copy new DNA or copy complementary DNA. So each strand acts as a template for the formation of new complementary DNA, forming a set of two DNA strands. So here we see on these further down, we see that this is the template on the one strand, and here we have the template on the other strand. And using the enzyme, and the enzyme in this case is DNA polymerase, that enzyme brings in the free complementary building blocks called nucleotides. So these templates are now exposed, we find that the nucleotides that are floating in the cytoplasm, the nucleoplasm, are brought in to base pair with their complementary strands on the DNA template. So the free nucleotides will move and they pair up with the exposed bases on the single strands. So here we've got these exposed bases, and so these free nucleotides will move in and they will pair up, which I've illustrated further down here. And this happens through an important set of enzymes called DNA polymerases. And this enzyme is able to match the bases, knowing that we've got adenine and thymine base pairing together, and guanine and cytosine always base pairing. So using that enzyme, you'll find that these templates will then serve as a template for the combination of a complementary strand. As this process occurs, the enzyme polymerase continues to read these strands, and it will help to base pair in the following order. So you'll know that a thymine will base pair with the cytosine, and likewise an adenine base pairs with the guanine. And as this process continues, polymerase continues to unzip the DNA, brings in more complementary free nucleotides to eventually form two new identical molecules of DNA, both of which are double-stranded. Each one will eventually twist to form a helical structure. So a simple illustration of the process would be that you've got a DNA molecule that then needs to unzip, and as it unzips, you're going to find that in the nucleoplasm, these templates now, which I'm going to enlarge in, okay, have on them the bases that are exposed. So if you have an adenine here, you've got a thymine there. If you've got a guanine here, you've got a cytosine there. And all of this is happening through the enzyme which is surrounding this, which is I've indicated as DNA polymerase. The DNA polymerase will help to bring in the complementary bases. And in this case, you've got adenine, thymine, guanine, and cytosine floating in the nucleoplasm that come in and base pair with their complementary templates to eventually form their complementary strands on either end. And as illustrated, you will get a strand. I'm going to go back to my red pen, okay, that now forms on each, forming two new strands that have double-stranded complementary DNA constituting them. So if we were to wrap this up in a few steps, the steps are as follows. The DNA unwinds, unzips. An enzyme called helicase helps to break the hydrogen bonds, as I've indicated in here. So this is the enzyme that's going to break them up. And this causes the two strands to separate, creating what we refer to as DNA templates. Okay? The second process would be that these templates form, and each strand acts as a template so that new complementary bases can come in and form. So here we're seeing that the new complementary bases are coming in based on the ATGC base pairing, that they start forming a complementary strand on the template. The next step would be the pairing up of the nucleotides, and we see here that these nucleotides start pairing up with that ATGC rule that we know and we've just illustrated, and they start eventually doing this with the help of an enzyme called DNA polymerase, and they start pairing up together with the free nucleotides floating in the nucleoplasm, and using that, the complementary base pairing occurs. And we finally see that once these base pairs are joined together with the combination of A and T combining, we look at finally the helix forming, which we see here, and the DNA starts reconfigurating into a double-stranded helical twisted molecule. And guys, that is essentially a wrap of how DNA replication takes place. So we've looked at the process, we've looked at the stages in which it occurs, and fundamentally important is the process of how it occurs is for you to be able to put down into a flow diagram and list the important processes of how the templates form, the enzymes bring in the free nucleotides to base pair with the complementary strands, to eventually form two new strands that are identical to each other, thus forming copies of the original DNA. Guys, you've done well. Let's take a little break, and when we get back, we will look at how the process of replication is important in terms of the cell division, and we'll try some application of this to some questions. Short break, and see you in a little while. Welcome back, Life Science learners, to our final installment. We've looked at the process of DNA replication. We've tried to unpack that process by looking at the different processes that occur to produce what we refer to as two identical copies. This process is a highly accurate process. There's significant proofreading of the DNA, which ensures that any mistakes that do occur when that template is being copied is corrected. We do know that these mutations do happen, and they often occur in more smaller organisms that have a really small genome that reproduce exponentially. So if we were looking at mistakes that do happen in the process of DNA replication, we refer to those as mutations. We're not focusing on mutations today, but it's important that we recognize that mutations do occur. But the rate at which they occur often takes us to the next process of trying to understand why is it that mutations do not occur as often as it would in the process of copying DNA. And so that takes us to the next process of looking at the models that explain DNA replication. And because of these models, we have a better understanding of why DNA replication is such an accurate process. So the first model that we're looking at is called the semi-conservative model. The second model is called the conservative model, and the third model is a dispersive model. And these models essentially try to explain the process of how replication can be looked at. An overview of these can be illustrated using simple colors. So right on the top here we have the semi-conservative model, and it produces two models of DNA as you see here. What is unique about these models is that if you look at this or these two new strands that are produced, each one of them has part of the original, which is in yellow, and part of a new, which is in blue. So we refer to this as a model showing half, which is semi, and conservative is referring to a term where we refer to keeping the old. So at the end of this process of replication using the semi-conservative model, we have two new strands, and each of these strands have a copy of the old strand and a new complementary strand. And so this is referred to as the semi-conservative model. The second model that we look at is the conservative model. And if you think of the word conservative, it essentially refers to keeping the old. And so the conservative model talks about DNA being replicated, where the original model is intact and a new copy of DNA is produced. And this strand is an entire new strand that is made up of entirely new produced or synthesized nucleotides put together into a form of DNA. And so scientists have rejected this purely based on that because of the frequency at which mutations occur, that this possibly is a model that will create a lot of faults in the DNA, essentially lots of mutations. A third model was also proposed, which was called the dispersive model. And this model talks about the new structures of DNA or strands of DNA having segments of old and segments of new dispersed within them. And so this model is found also to be erroneous, meaning prone to mistakes, and hence these two are the models that are not essentially accepted, whereas this model, the semi-conservative, is the one that is recognized as the most consistent model to explaining the accuracy of the process of DNA replication. So we refer to that as the semi-conservative, which produces new strands, each containing one of the original strand and one new strand. And so we're going to move on to trying to apply our understanding to this in terms of some questions. So let's get straight into these questions. The diagram represents a part of a molecule. Study the diagram and answer the questions that follow. So I'm going to call that diagram up in a bit. So here we've got a diagram of a DNA molecule that's illustrated. Let's try and understand firstly why this is DNA, and I can see that it's double-stranded. I also can see that there's possibly complementary base pairs, which I know that we will have to unpack in a little while. So that's an overview of that diagram. There's a key that's been provided, and that key again refers to the specific shapes and names of these nucleotides. So that's what's been given to us. Let's look at the questions based on this diagram. So the first question, A, identify the molecule in the diagram above. And so when I look at that molecule there, remember it's referring to the type of nucleic acid, and so that is DNA. And simply I can say that because I can see that it is a double-stranded structure. More important than that, we know that DNA would contain a thymine base, and we're going to try and find out if there's a thymine base present on that. The next question is label the parts numbered 1 and 5 respectively. So guys, it's important that you look at the question mark allocation. It requires that we label parts 1 and 5. It does not say 1 to 5. And so if you were labeling parts 1 to 5, it would mean that you would have spent an extra few minutes on questions that were not necessary. So it's important that you recognize that it requires that we label parts 1 and 5. So let's get back to this image. So we've got to label parts 1, and I'm going to identify quickly part 5. And so this would require us to be able to access the key. If we go to the key, we know that A represents adenine. So it means that if we look at adenine, which is clearly indicated at this position, it would mean that number 4 would be thymine. Okay? Using that, we know that again, that these bases would be, wherever you see that, there's going to be, let me get this right, a thymine there, and that's going to be an adenine based on its shape being similar to this base here. And we would know that if you've got guanine here, you've got to have cytosine complementary to it. And likewise, based on its shape and the similarity between this and number 5, we would know, and I can infer that number 5 has to be a cytosine base. So we look at number 1. Number 1 points to specifically the structures between A and T, and those would be the hydrogen bonds. So number 1 would point to the hydrogen bonds, and remember that these nitrogenous bases are held together by the hydrogen bonds. Okay, so that's what we're looking at for number 1. The next structure that we needed to identify was number 5, and I think it's specifically pointing to the skill of you being able to identify the type of nitrogenous base. Yes, number 5 is a nitrogenous base, but I think the question wants us to be very specific about the type of nitrogenous base, and using our inference that guanine and cytosine base pair, and looking at that shape, we can confer that number 5 refers to cytosine as the nitrogenous base. Cool. Let's move on to a few more questions. What is the collective name for the parts numbered 2, 3, and 4? So if we look at parts 2, 3, and 4, again, so I'm going to go to 2, 3, and 4. So we're going to put 2, 3, and 4 together. So 4 would be here. If we looked at them together, we know that a DNA nucleotide consists of 3 parts. It consists of a sugar, it consists of a phosphate molecule, as well as a nitrogenous base. So number 2 is the phosphate, number 3 is the sugar, and number 4 is the nitrogenous base. And so these 3 parts collectively are referred to as a DNA nucleotide. And we refer to that as a monomer of DNA, or a building block. Cool. The next question. What is the significance of this molecule being able to replicate itself? Now, the question refers to what is significant about DNA being able to replicate itself. And so if we understand the model of replication, and the model that we referred to was the semi-conservative model of replication, we can understand that simply that the structure of DNA with its complementary nature allows for free nucleotides to easily base pair when the strands are separated. So based on the unwinding and the separation into the templates, we know that these templates now form an easy access point for the free floating DNA building blocks to now base pair with their complementary nature. And so this talks to the structure of DNA allowing for the ease at which it can copy. So the significance of this molecule being able to replicate is that DNA is double-stranded. And so when the strands separate, each of these strands forms a template, allowing for complementary bases to base pair with these templates. And so it's important that we recognize that due to the double-stranded structure, as well as the complementary base pairs, that the process of DNA replication using the semi-conservative model of replication becomes efficient and a reliable method or model of replicating DNA. Right, so we'll move on to the next question. The diagram represents a portion of a nucleic acid. Okay, so here's a diagram that represents a portion of a nucleic acid. I'm going to try and read more into the question. Name the nucleic acid for 1 mark, and the second question is name 2 places in animal cells where this nucleic acid may be found. So let's try and identify which nucleic acid is illustrated in this image. If we look at that image closely, we can see that it's double-stranded. Again, that double-stranded nature of DNA is a distinguishing characteristic of DNA being double-stranded, and hence I use that as a significant point to be able to identify that DNA is the nucleic acid. Cool, that's a 1 mark question. Name 2 places in animal cells where this nucleic acid may be found. It's important that we recognize that firstly, we have DNA in the nucleus, so we refer to that as nuclear DNA or chromosomal DNA, so it can be found as chromosomal DNA or in the nucleus. But also we know that DNA can also be found in the mitochondrion. And if we look at the mitochondrion, obviously that's the cell, that's the organelle that controls the process of cellular respiration. And so DNA is found in there, and it's linked to providing the instructions to be able to produce important enzymes and controlling how the mitochondria function. The next part of the question requires us to identify different parts in this. So question C, identify portion V, W, Y. Okay, so let me read that again. We've got to identify portion V. We've then got to identify the nitrogenous base W, the molecule Y, and the bond Z. So a few things that we've read together. We're going to go back to this image and we're going to unpack each one of them. So if we look at V, and V again collectively points to all of these 3 structures together. And as I mentioned, if we look at these 3 structures, it's the 3 components that make up the building block of DNA. So V would be a DNA nucleotide. Okay? The next part that we need to identify is the bond Z. And as I mentioned that these bonds, which are the hydrogen bonds, hold the base pairs together. So that's your hydrogen bond. Okay? Y would be pointing to the phosphate molecule. So we know that DNA contains a sugar phosphate backbone. Okay? X would be pointing to the deoxyribose sugar, and it's fine for us to indicate that as a sugar, but it's actually pointing to this type of sugar, which is a deoxyribose sugar. And W in this case would be specific to what has been given on the one template. So that's a cytosine base, which would refer to this being a guanine nitrogenous base. And that is essentially the structures that we needed to label. The next question, what is the natural shape of this molecule? And due to the chemical bonding between the complementary base pairs, DNA takes on a helical shape. And so the natural structure of DNA is that it is helical. We move on to this last question. Name the process during which this molecule makes a copy of itself. And guys, we've spent the second half of this lesson today looking at the process of DNA replication. And so DNA replication is that process where DNA is copied to produce identical copies of itself. And we refer to that model as the semi-conservative model of replication. So guys, as we wrap up this section, a quick overview of today's lesson. We've looked at basically the structure of DNA as an overview. We've looked at the process of DNA replication. We've also identified the role of DNA in terms of as an important coding protein or coding nucleic acid in the cell. It's important that we understand that DNA replication takes place, when it takes place, and where. As a recap, DNA replication requires a complex series of enzymes that carry out this process to produce exact copies at the end. And this all happens so that the cell can prepare for cell division. So guys, you've done well, you've been focused. I hope that you've understood the process of replication. I encourage you to go back, read some of your notes, and try and make some of your own questions and answer them. So all the best, go well, keep safe.